Hi friends, today we are going to see the past year NEET questions for the chapter Strategies for Enhancement in Food Production. So already we have completed 4 parts of this same chapter. Kindly have a go through, so this will help you in the continuity with the questions. Now in this video we are going to do the part 5. So let's begin with the first question, which was asked in the year 1996. Suppression of reproduction of one type of organism by utilizing some features of its biology or physiology to destroy it, or by use of another organism, is known as dash. This question, as I already mentioned, was asked in the year 1996. What do you think will be the answer here? You need to understand the question first. You're suppressing the reproduction of one type of organism, either by utilizing some features, or you're doing, either it can be with biology or physiology. So what does it do actually? What is that called as? It is not competition. Competition is where one will be benefited, one will be affected. Predation is the same concept. Physiological control, no, we are controlling the reproduction aspects. Therefore, it is called as biological control. Therefore, the right answer is option C. Okay, next question here. What do you mean by rotinon? Okay, what do you mean by? Now, what do you mean by rotinon? And what is it actually? Okay, it is a natural insecticide with a minimal or almost no side effect. It is an isoflavonoid extract from the root of pteris. Okay, so what is it? It is a natural insecticide. If you want to know more about what is this pteris actually, maybe I can add some few points here so that you should not stammer. What is this pteris? See, they've said it is the pteris, root of pteris, right? See, this pteris is a genus of leguminous plants. Since they have mentioned it clearly, it's a root. So this is actually a leguminous plant which is found in the Southeast Asia and the southwest of Pacific Islands, including New Guinea. Now what happens here is they contain an important compound called as rotinon. Okay, they are a strong insecticide and a fish poison. Another detail that you may not know, here it is a fish poison, it seems. Okay, so you have many species which come under this. The one common one is the Pteris elliptica. Okay, fine. Next question here. One of the major difficulties in the biological control of insect pest is dash. You have to analyze the statements here. Option A, practical difficulty of introducing the predator to specific areas. Method is less effective as compared with the use of insecticides. Predator does not always survive when transferred to a new environment, and the predator develops a preference to the other diets and itself becomes a pest. So here, when there is a major difficulty in the biological control of the insect pest, what is the best way that you can do? What happens here is biological control refers to the use of the living organisms like natural predators and parasites to reduce the pest. Okay, so we can use natural ways where you can use all these methods to reduce the pest population. The main target is that for us. Okay, and they also, when the pest is controlled, of course the diseases can also come under control. So, however, they may become a pest and they can develop a preference to the other diets. Sometimes there are chances for it to turn the upside down, the result that we don't expect. There are chances for the predator to develop the preference to the other diets, and sometimes there are chances for them to become themselves as pest. Therefore, the right answer is option D. Next question here. Reproducing new plants by cells instead of seeds is known as dash. Very, very easy question. What do you think the answer is? We are not producing any seeds. We are producing directly plants, and that is called as tissue culture. Culture in the term generally used for the artificial growth. The plant tissue culture actually refers to the growth of plant cells, and you don't have any seeds there. It's directly cut and in artificial way it will be produced where you have the plants growing. Okay, so this is how you will do a plant tissue culture in an in vitro condition. Okay, therefore the right answer is option B. Next question here. Haploid plant cultures are got from dash. So where do you think you'll get this from? It's obviously not the leaves, root tips, and it should either be pollen grains and buds. All other are diploid. Okay, diploid in the sense they are 2n. Now they are asking us about N. Haploid plant cultures are got from pollen grain. Okay, so you can see a pollen grain development slide to your right side. Pollen grains have two coats. One is exene and the other is intine, an outer and inner layer. Pollen grains divide by mitosis, producing two haploid nuclei. So one will be a tube nucleus and the other will be the generative nucleus. Tube nucleus will form the pollen tube, while generative nucleus forms the male gametes. So out of the four options given here, leaf, root tip and buds are said to be diploid in nature, and only pollen grains are said to be haploid, and they produce two haploid nuclei. Okay, therefore the right answer is option C. Okay, therefore the answer is option C. Now let's move to the next question here. Most of our crop plants are dash. So what are the most of the crop plants are what? Either they are allo or heterozygous, mixed genotype, or autopolyploid in origin. When I say poly means many, we already discussed about it, right? Poly means many. Okay, the answer is most of the crop plants are autopolyploid in origin. Polyploid, which occurs within the species and results due to the multiplication of the same set of chromosomes, and that is called as autopolyploid. That is automatic. Okay, now you understand what is the definition for autopolyploid. That is the multiplication of the same set of chromosomes take place. That is why you call that as autopolyploid. So most of our crop plants are actually autopolyploid in nature or in origin. Which of the following plant cells will show totipotency? What do you mean by totipotency? Maybe we need to explain this. What is totipotency? See, totipotency can be defined or described as it is the ability of a single cell to divide and produce all of the differentiated cells of an organism. Spores and zygotes are examples of totipotent cells. The one which have the ability where a single cell will be able to divide and produce all the differentiated cells. So which one of the plant cell will show totipotency? It is the meristem region. Okay, xylem vessels or the cork cells are dead, while sieve tube cells do not possess nuclei. Therefore, the right answer to this question is meristem, which has the potency called totipotency. A single cell, okay, a single cell will be able to divide and differentiate. Okay, then that ability is called as totipotency. Therefore, the right answer is option C here. Next question here. Which crop variety is not due to induced mutation? Okay, which is not due to induced mutation. You will not know the answer here, but I'll tell you what the answer is. Rice, arar, wheat. The answer is option D, Arna of castor. Okay, it was genetically modified to get high lipid content. Okay, therefore the right answer is option D, and to your right side you can see a picture of how the fruits of the Arna of castor occur. Okay, next question here. Which of the following enhances or induces the fusion of protoplast? This question was asked in the year 2015. So what are the options that you have here? Polyethylene glycol, sodium nitrate, IAA, indole acetic acid, chitin, gibberellins, all these are growth factors, growth hormones that are given here. Now when you talk about the fusion, but before that, what is a protoplast? How can you define protoplast? What is its function? See, a protoplast is actually the protoplasm of a living plant or a bacterial cell whose cell wall can be removed. What is protoplast in biology terms? A protoplast of a plant or a bacteria or a fungal, it has the cell wall completely or partially removed using another mechanical or enzymatic mechanism. So what actually enhances or induces the fusion of protoplast is the polyethylene glycol and sodium nitrate, which is option A. Polyethylene glycol and sodium nitrate play an important role in the fusion of the protoplast of the same or different species. It is done for the formation of the somatic hybrid cell. This process is adopted from the normal sexual reproduction, and it is not possible for the production of hybrids. So this purpose, or this polyethylene glycol and sodium nitrate, they are used for fusion process. It will not help or it will not induce any hybridization method. Okay, therefore the right answer is option A. Next question here. A technique of micropropagation is dash. Very easy question, a direct question also. What is the answer here? Why? What is micropropagation? What does a micropropagation do? Maybe we need to discuss that first. See, a micropropagation is a technique, okay? It is where the propagation of plants by growing plantlets in a tissue culture and then planting them out. So it's a practice that is rapidly involved for multiplying the stock plant material to produce as many progeny plants. So a technique of micropropagation, I've given you the clue. The right answer is somatic embryogenesis. Okay? Yeah. I told you we are actually multiplying the stock. The main technique, I mean, why are we following this micropropagation? The main thing is it is a practice rapidly to multiply the stock plant material to produce as many progeny plants using modern plant tissue culture methods. It is used to multiply the plants such as those which have been genetically modified or bred through conventional plant breeding methods. Okay, next question here. To obtain virus-free healthy plants from a diseased one by tissue culture technique, which of the diseased plant can be taken? Okay, so in this situation, whether you will take a palisade, apical or epidermis, to obtain a virus, a free healthy plant from a diseased one by the tissue culture technique, you have to actually take both. You actually have to take both, that is the apical and the axillary part. Okay, plant tissue culture is a method that is used to maintain or grow plant cells, tissues or organs under sterile conditions with a supplement of enough nutrients. Okay, and that too, when you supply nutrients, it will be in the right composition. Plant tissue culture is widely used to produce clones of the plants by the method called as, or simply called as, micropropagation that we saw in the earlier question. So whenever we to obtain virus free healthy plants, we take in the both apical and the axillary meristems. Okay, next question here. Which one of the following has the maximum genetic diversity in India? Very easy question. What is the answer? Yes, it is rice. During the period of 1960 to 2000, rice production went up to 35 million tons to 89.5 million tons, it seems. So this was due to the development of the semi-dwarf varieties of rice. And now you have about 2 lakh varieties of rice in India. Therefore, the right answer to this question is option A. Next question here. Tissue culture technique can produce infinite number of new plants from the small parental tissue. That's right. The economic importance of this technique is to raise what? You have to analyze the equation, the statements here. Variance through picking up a somoclonal variation, wrong. Genetically uniform population identical to the original plant, that is the right answer. So why do we undergo this tissue culture? One is to preserve the original state or the parent, and we have to, and the production, the demand for the production also is high. So when we talk about these things, the method of producing thousands of plants through tissue culture, as we all know, what is that technique called as? Micropropagation. Right, it's called as micropropagation. Each of these plants will be genetically identical to the original plant from which they are grown. That is, they are actually somoclones. But the exact answer is this, option B, genetically uniform population, which is identical to the parent, original parent. Okay, next question here. Which part would be the most suitable for raising a virus-free plants for micropropagation? Out of the 4, the right one is meristem, which is a haploid set of chromosomes. And meristem, if you see, is the most suitable for raising a virus-free plant, and especially when you go for an artificial method of tissue culture. Therefore, the right answer is option C, meristem. When you talk about meristem, you also have shoot meristem, root meristem. It's called shoot apical, root apical. Apical meristem causes primary growth, while lateral meristem causes secondary growth. To your right side, you can see a clear image of it. Therefore, the right answer to this question is option C, meristem. Next question here. Which one of the following is a case of wrong matching? You need to analyze the statements here. Somatic hybridization is a fusion of 2 diverse cells. You have to get the wrong one. Okay, vector DNA is the site of tRNA synthesis. That is wrong. Vector DNA are the DNA molecules that carry a foreign DNA, right? They carry a foreign DNA segment and replicate inside the host cell. Vector DNA that we mostly use are actually plasmids. What are plasmids? They are extracellular circular chromosomal material, right? So the wrong statement out of the 4 is option B. Vector DNA is not the site for tRNA synthesis. It is actually a foreign material that is carried and it is made to replicate into the host cell. Therefore, the right answer to this question is option B. With this, we complete the part 5 of this chapter, which is strategies for enhancement in food production. Hope to meet you in the last part of the same chapter in the next video. Thank you.